Okay, hello everyone. I hope you have gone through my last lecture, or say as the last video. So this is the, I should say, the final video of our chapter number 11, that is biotechnology principle and its process. Okay, so before starting the video only, I am giving you the instruction that please take a notebook and note it down, because in this video we are going to see the whole process of our DNA technology, and many special neat questions, or say has the neat queries would be solved over here. So please take a note and note the points. So let's start. So today we are going to see that is 11.3, that is processes of our DNA technology. Okay, I know that from the last few videos we are going through the DNA technology only, but from the in this video we are going to see detailedly the whole process of our DNA technology, that is recombinant DNA technology. So there are several steps for recombinant DNA technology. Okay, first of all, that is the isolation of the DNA. Second one is fragmentation of DNA by RE enzyme, that is restriction endonuclease enzyme. Third one is separation and isolation of desired DNA fragment. Fourth one is amplification, that means multiplication of our gene of interest. The next is ligation of alien DNA into vector. After ligation, that is the transformation process, and after that culturing the host cell at large scale. It means that in the industrial scale, and extraction of the desired product. So first of all, we will start with the isolation of DNA. As you know that nucleic acid is the genetical material of all living organism, and it is present inside the cell. So first of all, we have to break the cell membrane. Of course, right. Why we have to break the cell membrane? Because DNA is present inside it. So as you know that all cell membrane in different organism is composed of different material. So our target is to break it. And so for that, if it is a bacterial cell, see over here, if it is a bacterial cell, we would use the lysozyme enzyme. If it is a plant cell, then we will use the cellulase enzyme. And if it is the fungi, or say as the fungi, then we would use the chitinase. As I mentioned earlier, ASE stands for the enzyme. So wherever you see the last three letters, that is AXE, say as the suffix as ASE, then it is an enzyme. So first of all, for isolation of the DNA, we need to break the membrane. For that, we would use the specific enzyme for the specific organism. After using the enzyme, now after using the enzyme, say for example, now I will say that now as the cell has been broken, so along with the DNA, cell also contain the other macromolecules such as the RNA, protein, and out of this our target is to isolate the DNA. So for this purpose, we have to eliminate RNA and protein, right? So for this, for the elimination of the RNA, we would use the ribonuclease enzyme, which would eliminate, or say as which will degrade the RNA. And for the protein, to eliminate the protein, we would use the protease enzyme, or say as for degradation, we would use the protease enzyme. So now ultimately, the left out material which we have is DNA. Now this DNA is being precipitated using the, see over here, using the chilled ethanol, which after precipitation will appear as a fine thread. This is an extra point, that when we use the chilled ethanol, our DNA would be precipitated as the fine threads. Okay. Now second point means here we have done the isolation of DNA. After isolation, what we will do? Fragmentation of DNA by restriction endonuclease enzyme. So now from isolation we got the whole DNA, out of which we need to cut our gene of interest, right? So for cutting, what we use for that? That is restriction enzyme. So for that purpose, the purified DNA had to be incubated at proper conditions. See over here, second one, that is incubation at the proper condition, which are specific for the specific enzymes. Okay, first of all, what we have done? We have done the isolation of the DNA. For that, हमने क्या किया था? हमने सेल मेम्ब्रेन को रिमूव किया था फॉर स्पेसिफिक लिविंग ऑर्गेनिज्म, स्पेसिफिक एंजाइम्स आर यूज्ड फॉर द रिमूविंग द सेल मेम्ब्रेन. आफ्टर दैट हम वी विल गेट द मैक्रोमोलिक्यूल्स. मैक्रोमोलिक्यूल्स क्या होता है? वी विल बी गेटिंग द डीएनए, आरएनए, प्रोटीन. हमारा टारगेट क्या था उसमें से? Our target is just the DNA. So we have to eliminate RNA as well as protein. तो RNA और protein हम कैसे eliminate करेंगे? With the help of ribonuclease enzyme and the protease enzyme. ये होने के बाद जो DNA material है उसको हमें precipitate करना होता है. So for the precipitation process we will be using the chilled ethanol. जैसे ही chilled ethanol डालेंगे, precipitation कैसे होगा? हमें जो DNA material है वो हमें full fine thread के form में दिखा जाएगा. After that, our second one is fragmentation of DNA by RE enzyme. इसमें हमने क्या देखा था? We have to incubate at the proper condition. Now third one, that is separation and isolation of desired DNA fragment. अब हम separation कैसे करेंगे? And how we can isolate? So मेरे previous lecture में एक electrophoresis का portion था which I told that I would cover up in the next my video. So this is the video for that.